Hello everyone, welcome to EPG Pathshala. I am Dr. Surbhi Gupta. I am an assistant professor in the Department of Reproductive Biology at All India Institute of Medical Sciences, New Delhi. Oocyte, structure and biology. The objectives for this module will be, we will discuss the structure of the mammalian oocyte, then we will talk about the oocyte production and maturation both during the fetal life and childhood and also during the adult life. Then we will discuss the functional attributes of the oocyte, which will include fertilization, cortical reaction, zona hardening, polyspermy block and also oocyte activation. So, we all know that small organisms like amoeba etc., they produce asexually, which means they just divide by fission and that is how they reproduce. But higher organisms, they undergo sexual reproduction which needs gametes. So, why are gametes important? Gametes are important because when two organisms, higher organisms are there, they have what is called 2n number of chromosomes in their each cell, but the gametes, they are specialized cells which have only half the genetic material. This halving of the genetic material is necessary to ensure that when fertilization occurs, both the gametes combine and give their half or haploid set of chromosomes or genome, and the resulting zygote results in 2n or the complete set of genome. This leads to the formation of a multicellular organisms after mitosis, which keeps the chromosome number constant, and then again the cycle repeats because after meiosis the zygote again, then the process repeats itself because after meiosis the gametes again they have only haploid set of chromosomes. If this process did not occur, then what would happen is that each resulting generation would have double the amount of genetic material or DNA or the number of chromosomes, and with each successive generation the genetic material would keep becoming double. To ensure this, gametes are created in higher organisms which gives only haploid set of chromosomes from the paternal side as well as from the maternal side, and thus after fertilization the complete set of chromosome which have the same number as the maternal and paternal parent is formed. So, in most higher organism there are two kinds of gametes, one which comes from the maternal side which is called the female gamete or the oocyte or the egg, and the paternal gamete or the sperm. Both the gametes, they differ highly in their morphology. The oocyte in most cases is of a large size and is usually immotile, whereas in all cases sperm is highly compact and highly motile. This is because the oocyte needs large energy reserves so that they can be used when the zygote develops. So, during early development of zygote and embryo, this energy reserve which the oocyte has will be required. But the compact size of the sperm and the high motility of the sperm is required so that the sperm can reach from the paternal parent into the maternal body and affect fertilization. As you can see here in the picture, the typical mammalian sperm is about 2000 times smaller than the mammalian oocyte. This is what a typical mammalian mature oocyte looks like. Usually it is about 100 to 140 micron in diameter. It depends on the species what is the size of the oocyte. Consists of, like all other somatic cells, it has a plasma membrane, it has a large nucleus which contains all the DNA or genetic material which is required for passing on the genetic information to the offspring. It has a big cytoplasm which, as I just mentioned, has all the energy required for development of the zygote or the embryo. Usually the first polar body which is formed after meiosis is retained within the zona pellucida, which we will talk about later, and also it has cortical granules around its cytoplasm just beneath the plasma membrane, and then the zona pellucida is surrounded by a group of cells which are the follicle cells or the cumulus cells. These we will see how they are important for development of the oocyte. So, the first objective of this module is to discuss the structure of the mammalian oocytes. So, the oocyte or the egg is similar to all somatic cells in terms of the essential organelles that it has: the plasma membrane, the nucleus, the mitochondria, the ribosomes, and all the Golgi apparatus and all other similar organelles. However, there are a few features which are specially present only in mammalian oocytes. One of them is the zona pellucida. The zona pellucida is a translucent acellular layer which surrounds the plasma membrane of the oocyte. Like you can see in the picture here, the middle picture shows a diagrammatic representation of the mammalian oocyte, where the light colored membrane surrounding the plasma membrane of the oocyte is the zona pellucida, and the picture on the left shows a close view of the zona pellucida, how it has cross-linking polymers which make it up. And on the right hand side is the picture which shows the actual photograph under a microscope of a mammalian oocyte. You can see in this picture the cytoplasm of the oocyte, and on one side is the polar body, which is both of which are surrounded by the acellular zona pellucida layer. The zona pellucida is a special form of extracellular matrix which surrounds the oocyte, and it separates it from the surrounding granulosa follicle, or in other name of it, the cumulus cells. However, some cytoplasmic connections pass through the zona pellucida which connect the oocyte with the cortical cells. However, some cytoplasmic connections pass through the zona pellucida connecting the oocyte to the cumulus cells. This zona pellucida is made up of glycoprotein molecules which are secreted by the oocyte itself during oocyte development. The cytoplasmic processes which pass through the zona pellucida are important as they form gap junctions, and through these gap junctions important macromolecules can pass through, which are sometimes required by the oocytes for its development. The presence of the zona pellucida is not only required during oocyte development, it is also required during fertilization, because it has to, after fertilization, it has to protect the developing zygote or embryo from the outside environment. In mammalian fertilization, the fertilization is internal or inside the body. However, in other species, in lower species like birds and reptiles, after fertilization, the egg has to be surrounded by something which can protect it from the outside environment. The human zona pellucida is made up of 4 glycoproteins which are named as VP1, VP2, VP3 and VP4. VP1, VP3 and VP4 bind the capacitated human sperm and allow it to induce the acrosome reaction, whereas VP2 can bind only acrosome reacted sperm. So, this ensures that only sperm which have undergone all the maturation process can bind and fertilize the oocyte. The Cellullar-Lewis X sequence, which is present on both N and O glycans of the human zona pellucida, are important for sperm binding. The N-linked glycans on the human VP3 and VP4 are responsible for inducing the acrosome reaction. In mice, there are only 3 glycoproteins which make up the zona pellucida, namely VP1, VP2 and VP3, and in both cases VP2 is responsible for binding acrosome reacted sperm. The other special feature of mammalian oocytes are the cortical granules. They are present as secretory vesicles just inside the plasma membrane in the outer region of the oocyte cytoplasm. They are distributed evenly throughout the cortex region except near the pole where the nuclear and the first polar body are usually present. There is a reason for this, which will be discussed later. The cortical granules are similar to the acrovome present on the sperm, that is, they are produced by the Golgi apparatus and they have many proteolytic enzymes which are required for Wona reaction. The second part of this module, we will talk about how the oocytes are produced and how they mature. So, the process of oogenesis or oocyte production requires three kind of steps. One is the proliferation, which happens by mitotic divisions. The second is the genetic reshuffling and reduction in the number of chromosomes, which is by meiosis process. And the third, the maturation step or the cytodifferentiation of the oocyte to become a functional mature oocyte. This picture shows the different stages of oogenesis. As we can see here, the initial oocyte germ cell is called the oogonia, which has 2n number of chromosomes, that is the complete diploid set of chromosomes. With meiosis, it leads to additional oogonia. However, this mitosis process is restricted only in the fetal period of a human female. By the time the human female is born, all the oogonia have undergone first stage of meiosis and have formed primary oocytes. These primary oocytes cannot multiply and they are arrested in this stage at the time of birth. When maturation has to take place, this primary oocyte divides into the secondary oocyte and the first polar body. After this, the secondary oocyte again undergoes division and forms the mature oocyte and the second polar body. In some cases, the first polar body divides into two polar bodies, but that may or may not happen in an individual. The second oocyte gives rise to the mature oocyte, which fertilizes and forms the zygote. During early embryogenesis or in the fetus, primordial germ cells arise from the epiblast layer and they form the oogonia. They move from the yolk sac to the genital ridge where the mature gonads will be formed. During the migration, they divide by mitosis, and by the time they reach the genital ridge, they increase in number to about 5,000. At the same time, some other cells migrate to the same position and form small clusters around these primordial germ cells. At this stage, the cells in a female are called oogonia, which are consisting of the germ cells in the center surrounded by small clusters of cortical cells. These oogonia divide by mitosis and ultimately enter the meiosis process and form the primary oocyte. By the time a female is born, all the primary oocytes have been arrested in prophase 1 of meiosis, and this is the number that the female will ever have during her reproductive cycle. In contrast, if we see the spermatogenesis process, the mitotic potential is maintained till the end of the life and the number of spermatocytes that a male can produce is not restricted. The primary oocytes are arrested at diplotin stage of prophase 1, but all the chromosomes are enclosed in a nuclear membrane known as the germinal vesicle. These arrested oocytes are said to be at the dictate stage where they will remain until the female reaches puberty. At the time of birth, the ovary contains about 1 to 2 million oocytes. However, a majority of these degenerate before puberty and through the reproductive life of the female. By the time the puberty is reached, only about 3000 primary oocytes are remaining. So, to summarize the timeline for the PGCs, in the second week of the fetal development, the primordial germ cells are formed in the epiblast and they move to the wall of the yolk sac. By the fourth week, they have migrated from the yolk sac to the developing gonads region. By the fifth week of development, they have reached the final position at the urogenital ridge. By the third month, they have multiplied by mitosis and the maximum number of oogonia are present at this time point. From the fourth to seventh month of the fetal life, the primary oocytes are formed by prophase 1 of meiosis, and by the time of birth in a female, they are arrested at diplotin stage of prophase 1. So, this is showing how the number of germ cell changes over the life period of the female. As you can see, the maximum number is seen during the second trimester of the fetal life, whereas by the time puberty is reached, only a few primary oocytes are remaining, and that also keeps declining as the reproductive lifespan is, and that also keeps declining as the reproductive life cycle goes on. With the onset of puberty, a regular recruitment of these primordial germ cells takes place. The initial part of the growth happens majorly in the primary oocytes. They increase in diameter to attain their final size of 60 to 120 micrometer. Though the oocyte is still arrested in the meiosis, the chromosomes are busy in synthesizing huge amounts of ribosomal RNA and messenger RNA. These messenger RNAs code for all the building material required to synthesize organelles and also to generate the store of proteins which will be used later during the maturation of the oocyte and also during the first few days of embryonic development. During this period only, the oocyte secretes materials from which the zona pellucida is formed. This maturation of the oocyte takes place after puberty. During each reproductive cycle or during each menstrual cycle, some oocytes are taken up by the maturation process and they increase their size. However, till the LH surge happens or the luteinizing hormone surge happens, the chromosomes are contained in the germinal vesicle or the nuclear membrane in the cytoplasm. After the LH surge happens, the chromosomes complete the meiosis 1 and the first polar body is relieved. Half of the chromosomes go to the first polar body, whereas the other half are retained in the oocyte. Interestingly, the cytoplasmic division which takes place along with this division of the chromosomes is unequal, resulting in a small polar body and a large oocyte. This ensures that all the materials required for later zygote development are stored in the oocyte itself and are not lost in the polar body. After the completion of meiosis 1, the oocyte immediately enters meiosis 2. At this stage, they are called the secondary oocyte. However, the moment they enter meiosis 2, they are again arrested, and this arrest is relieved only after fertilization in most mammals. When they are arrested in meiosis 2, they are released from the ovary by the process of ovulation and they enter the fallopian tube where fertilization will take place, provided sperm are in the vicinity. This slide shows how the oocyte matures from the GV stage or the germinal vesicle stage to the metaphase 2 stage. As you can see, the germinal vesicle or the nuclear membrane which surrounds the chromosome is in the center when the oocytes are arrested in the first meiosis. When the luteinizing hormone surge happens, the chromosomes start moving towards the pole, at which point the chromosomes separate and the first polar body is extruded. The first polar body is extruded close to the pole, at which location there are no cortical granules, As I had mentioned earlier, the cortical granules are not present here because this area is required for formation of the spindle assembly. And you can see that after the first polar body has been excluded, the metaphase 2 stage oocyte still has the chromosome close to the polar body, and the spindle assembly is still arranged here. This slide shows an actual photograph of the oocytes at different stages of maturation. The first picture on the top shows the germinal vesicle stage oocyte. You can clearly see here the big nucleus around which the whole cytoplasm is there. The second picture on the top shows the metaphase 2 stage oocytes where the first polar body has been expelled. So, you can see the nuclear membrane has broken down and one cannot make out where the chromosomes of the secondary oocyte are present. However, the location can be approximated based on the location of the polar body. The lower part of the picture shows a close-up view of the eggs at different stages, the metaphase 1 and the metaphase 2 stage. One interesting thing which needs to be pointed out is the difference in the oogenesis and spermatogenesis. Both the process, the production of the oocyte and the sperm, happen by meiosis. However, the process of spermatogenesis leads to the production of 4 haploid sperm which are all equal, whereas from one primary oocyte, only one mature oocyte is produced during the process of oogenesis, because all the other 3 become polar bodies. Let us move on now to the functional attributes of the oocyte. The most important function of the oocyte is fertilization. Fertilization means the fusion of sperm and egg plasma membrane. There are special regions of both sperm plasma membrane and oocyte plasma membrane which are involved in the fertilization process. The equatorial segment of the sperm plasma membrane is the site where fusion takes place. Similarly, the polar region of the oocyte plasma membrane is not involved in the fertilization process. The other portion of the oocyte plasma membrane is full of microvilli, and it is with these microvilli that the sperm membrane fuses and the cytoplasm of the sperm combines with the cytoplasm of the oocyte. Specific molecules are required on the surface of both sperm and egg for fertilization to take place. Studies which have involved generation of gene knockout mice have shown many molecules which may be involved. Currently, 2 which are known to be present are ISMO on the sperm surface and its receptor Juno on the egg membrane. Other protein molecules like integrins on the egg surface and tetraspanin molecules like CD9 and CD1 also play important role in the fertilization process. However, their direct role in fertilization has not been shown, and it is believed that binding of ISMO protein on the sperm and Juno on the oocyte surface brings the membranes of both the gametes closer, which helps in merger of the lipid bilayers into a single lipid bilayer. Once fertilization takes place, the cortical reaction takes place. The cortical reaction is very important for prevention of polyspermy, because we all know that if too many sperm enter the oocyte, the resulting zygote will have more than the diploid set of chromosomes, and that will be detrimental for embryo. The cortical reaction takes place in the cortical granules which are produced by the Golgi apparatus. They contain the proteases which after fertilization are relieved. These proteases released by the cortical granules react with the zona pellucida and change the glycoproteins of the zona pellucida. This leads to hardening of the zona pellucida and block of further sperm entry. The picture here shows how the zona pellucida is changed during fertilization. Before fertilization, the sperm can bind to the zona pellucida proteins VP1, VP2 and VP3, whereas after the cortical reaction takes place, the VP2 molecule and VP3 molecule are modified. The VP2 is cleaved and it is no longer able to bind to the sperm. The cross-linking between VP1 and VP3 also changes and the zona is hardened. This is very essential to ensure that only a diploid set of chromosome will be present in the developed embryo. Here is the diagrammatic representation of the polyspermy block. The zona pellucida, as we mentioned, is composed of the cross-linked polymers of VP1, VP2 and VP3. After the cortical reaction takes place, the VP2 is cleaved and the VP3 glycoprotein is also modified. Because of this, the sperm are not able to attach to the VP molecules, and hence the first sperm is the only sperm which will be able to fertilize the oocyte. Fertilization of the sperm leads to activation of the oocyte. A increase in the intracellular calcium of the oocyte is very important for this oocyte activation to take place. There are many theories of how sperm activates the oocyte. One important one is that it has a protein called PLC-zeta, phospholipase C-zeta, which passes from the sperm into the oocyte cytoplasm at the time of fertilization. This leads to activation of the IP3 gated channels on the endoplasmic reticulum and releases calcium ions. The calcium ions in the cytoplasm, they act on the cortical granules and allow it to release their content, because of which zona hardening takes place, which we have just discussed. Another theory says that the increase in the intracellular calcium, which leads to oocyte activation, could be because there is a direct entry of intracellular calcium from the sperm into the egg following fusion. The other hypothesis is there is a release of sperm-bound oocyte activating factors into the egg, which cause this increase in IP3 mediated calcium release. Oocyte activation leads to not only completion of the female gamete meiosis, that is extrusion of the second polar body, but also it is required for decondensation of the paternal chromatin, because the sperm chromatin is highly condensed so as to minimize its size. So here the proteins which were bound to the DNA in the sperm are relieved. They are replaced by the histones present in the oocyte and the sperm DNA becomes decondensed. This leads to formation of the male and female pronuclei, and ultimately they come together at the equatorial plate. The mitotic spindle is formed, and this is the point at which the oocyte is ultimately formed. So to summarize, we have the oocyte, which is the female gamete, which is large in size because it needs to be the energy store for the developing zygote and embryo, whereas the sperm has to be motile to reach the oocyte to fertilize it. The two important structural features of the mammalian oocyte are the acellular layer of zona pellucida and the cortical granules, which are very important to prevent the polyspermy to happen. The production of the oocyte or oogenesis happens in stages, during which there are many arrests which happen. At the time of birth, a human female has all the primary oocytes that she will ever have, which are arrested at the prophase 1 of meiosis. Then they get activated with each menstrual cycle after puberty, and the secondary oocyte is again arrested at meiosis 2 stage. It remains in this arrested state till the sperm activates the oocyte and the second polar body is extruded. This happens only after fertilization. After fertilization, there are many other changes which happen, like cortical granule reaction, zona hardening and oocyte activation, which leads to formation of the male and female pronuclei and ultimately the zygote. That is the end of this module. Thank you.